Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Rapid global urbanization has dramatically changed the face of our planet, polluting our atmosphere with greenhouse gases and causing global warming. It is the need of the hour to control our activities and find more sustainable alternatives to preserve what remains of our planet for the generations to come. Carbon dioxide (CO2) and carbon monoxide (CO) make up a large proportion of industrial flue gases. Recent research has shown that certain microorganisms are capable of metabolizing these gases into useful byproducts. Thus, attempts are now being directed to using microbes to recycle these gases and convert them into useful chemicals in a process known as carbon capture and utilization (CCU). This is a step beyond the current widespread practice of carbon capture and storage (CCS). However, such CCU requires high energy input, making the scaling up of this process difficult and expensive. How can this process then be optimized for maximum output? A team of researchers from Korea, led by Professor Young-Rae Kim from Pusan National University, have answered this question for a newer CCU system called the Bioelectrochemical System (BES). Professor Kim explains, "We have developed a bioelectrosynthetic process in which electroactive bacteria convert CO2 into useful metabolites like acetate and volatile fatty acids using electricity as the reducing power." The scientists were able to optimize BES to increase their efficiency by 2t. Their findings are published in Bioresource Technology since January 2021. The two-chamber BES they used had several special features that achieved this. The cathode contained an electroactive biofilm, and the anode produced hydrogen ions via water electrolysis. These chambers were divided by an ion exchange membrane (IEM), which controlled the flow of protons and electrons between the chambers. Further, while the former contained microbial culture media, the latter contained mechanisms to control the initial pH of the system. In addition, a quinone electron mediator was used. They found that, given the right IEM, one that allowed protons but not oxygen to pass through, an acidic pH in the anode chamber caused a higher proton concentration gradient across the membrane, which was key to enhancing acetate production and the synthesis of longer-chain fatty acids in the quinone-dependent mediators improved electron transfer and increased product formation. Professor Kim states, since CO is a more reduced gas than CO2, perhaps unsurprisingly, the Coulombic efficiency for CO was double that for CO2. CO is a major component in the industrial off-gas of most steel mill processes and biomass gasification. Via this BES conversion, it can be a valuable feedstock for various bioprocesses. This is the first study that makes the conversion of CO via BES commercially viable. Highlighting the applications further, he continues, the microbes self-replicate, making this BES an economical solution. That combined with the efficiency we've achieved and the optimal system we've created should make this of sufficient interest to industries such that this becomes commercial industrial machinery within 5 years. Story source: materials provided by Pusan National University. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.